Hello, this is lesson 9 from the B6 inheritance topic, and it's all about cloning. And this lesson is only for our triple biology students. So if you're studying combined, you can miss this lesson out and move straight on to lesson 10. My name's Mrs. Keenan and I'm Science Director for Outward Grange Academies Trust. And we're going to start off by thinking about what we mean by the term cloning. How might we use it? Can you think of any examples? And then a bonus mark if you can identify who it is in the picture. So I'm just going to give you 30 seconds just to think about those ideas and just get our brains working. Okay, time's up. So, the term cloning. You might have heard about Attack of the Clone or the word clone in films or on television as being something that's identical to the original thing. So if you were to make a clone of yourself, you would make an identical copy of yourself and it would be exactly the same as you in every way because it would be genetically identical. We might use cloning to stop species becoming extinct. We might use it to reproduce something of a high quality very quickly in a high number and make more money. I'm thinking about plants or commercial gardens or farming. And the examples you may have come up with may be cloning some simple plants, but I'm not sure you'll have known beyond that. You might have heard about cloning when we did about asexual reproduction. And asexual reproduction is the type of reproduction which produces clones, and those clones are genetically identical to the single parent that produced them. And then I said to you there was a bonus mark for identifying the sheep in the picture is Dolly the sheep. She was the first mammal ever to be cloned from an adult cell. So when she was cloned, it was actually fairly breakthrough news. It was all over the BBC News, the international news, because we weren't sure if we would ever be able to clone a large mammal. But when Dolly the sheep was produced, there were all sorts of assumptions being made that we could now start cloning humans. And some people thought that was really exciting. And some people had real grave concerns about this as being science gone mad. So Dolly the sheep was cloned in 1996, so 24 years ago, and she was the first mammal ever to be cloned from an adult cell. And later in the lesson, you'll understand a little bit more about what I mean by that term. Okay, our outcomes for today. Our challenge outcome, we are going to describe the differences between sexual and asexual reproduction. And our Aspire outcome, we're going to evaluate all the different methods of cloning. So, just to get us thinking then, I want you to look at those two pictures and I want you to decide what are the differences in reproduction in these two pictures. And I'm just going to give you 30 seconds again. Okay, so on the left, we've obviously got this cartoon image of your typical family, mum, dad, two siblings, boy and a girl. If we are referring to people, humans, we are looking at sexual reproduction because there's two parents, there'd be a mix of gametes and the offspring would have variations. The only exception to that is in identical twins, when just one sperm fertilises one egg and that developing embryo splits in the early stages of development. but generally humans reproduce by sexual reproduction. The picture on the right shows these large field daisies. They can reproduce asexually. So all of the daisies would be genetically identical. They would be clones. Similarly, many plants can reproduce sexually by having pollen transferred from one plant to another, often by insects such as bees. And some plants can reproduce by both asexual and sexual reproduction, depending on the conditions that they live in. So 2 types of reproduction, just a reminder from a lesson that you studied earlier on in this topic. Humans reproduce by sexual reproduction. This is the fusion of male and female gametes. And remember, gametes are just sperm and egg. The mixture of the genetic information from the 2 parents leads to variety in the offspring. And if anybody's got any gaps in their knowledge about sexual reproduction, it's the first type of reproduction you need to know about. You can pause the video now and you can make a note of those key points. Asexual reproduction then is the way that some plants reproduce. If it's asexual reproduction, there is no fusion of gametes, as there's only one individual needed as the parent. So therefore there is no mixing of genetic information and no genetic variations in the offspring. So the offsprings would all be genetically identical individuals, which are known as clones. And again, if you've got any gaps in your knowledge about asexual reproduction, which is the second of the 2 types of reproduction you need to know about, pause the video now and make a note of those key points. Okay, looking at today's lesson then, we are going to look at different methods of cloning. And the first method of cloning we're going to look at is when you take cuttings from plants. So a plant can easily and quickly be cloned by taking a cutting. This is a fast way of cloning a plant. So all you do is you take a bit of the plant, you cut it off, it might be one of the side branches, stem. You take that bit of plant, you dip it in some rooting powder, which would contain the hormone auxin that will promote growth in the plant, and you plant it into some compost. You might decide you're going to cover it with a plastic bag to retain moisture just for the first few days until that cutting gets going. This method is quick, it's cheap, but we can't produce many clones quickly. Gardeners do it with strawberry plants. They send out side branches and you can just chop them off and put them in new compost. Those strawberry plants produced would be genetically identical to the plant they were taken from. There's also a plant called a spider plant, which sends off these side branches and then they develop into perfectly identical plantlets or small plants if they're cut off and they're planted in compost or soil. So taking cuttings is a very quick and a very easy and a very cheap way of cloning plants. The second method of cloning that you need to know a little bit about is tissue culture. Now, tissue culture is similar to taking a cutting, but it's a little bit more complicated. In tissue culture, the tissue sample is taken from the parent plant. So in the same way as the cutting was taken from a parent plant, a very small amount of tissue sample would be taken. That tissue sample would be placed in agar growth medium, which contains nutrients and auxins. So you might have used these Petri dishes with this agar jelly on it when you've done about when you've looked at microbiology and you've grown bacterial samples. Similar kind of growth medium would be used, but this time it would contain the nutrients and auxins to ensure that those tiny little bits of tissue sample from the plant do then grow and develop into tiny little plants called plantlets. And again, the lid would be put on to just retain the moisture, stop them drying out until they get going. Once those little plantlets have developed, they would be separated out onto a compost tray and allowed to develop into individual plants. Again, they would all be genetically identical to the parent plant, but the advantage of tissue culture is you can produce a high number of plants from one plant, but it is more expensive because there has to be a laboratory intervention in terms of the petri dishes and the nutrient medium, and the samples taken rigidly, and you would need to use a microscope to check that you're actually getting the right bit of the plant. So it's more expensive, but does produce a higher yield of plants at the end. So I'm going to give you 3 minutes, and I want you, just on your piece of paper or in your books, to describe the 2 methods of cloning in plants, starting with cuttings, moving on to tissue culture, and I'll let you know when half your time is up and you should have the first one finished. 3 minutes starting now. Okay, so over half your time's up. You should be now on tissue culture. Okay, time's up, brilliant. So for the cuttings, you should have talked about taking a small bit of the plant, cutting it away from the parent plant, planting it in compost, and using some growing powder or hormone auxin or rooting powder to make sure that plant actually does develop properly. For tissue culture, you should have spoke about getting a sample of tissue from the plant, planting it into an agar dish with nutrient agar, which contains auxin and hormones and nutrients, and then allowing that tissue sample to develop into tiny plantlets in a petri dish. And once those plantlets have developed, transplanting them into a compost tray so they are able to grow independently. For tissue culture, you should have written about lots of offspring plants will be produced. For cuttings, there's a smaller number, but for cuttings, it's cheap and it's easy and it's quick. And for tissue culture, it takes longer, it's more expensive, but it does actually produce a higher number of plants in the end. So just a reminder of the diagrams for the cuttings and the diagram for tissue culture. Very similar process, but tissue culture slightly more complicated, would cost more, but would produce a much higher yield of plants at the end. Okay, so just to summarise then, I would like you to produce a table that outlines the advantages and disadvantages of these two methods of cloning plants. So one table which shows the advantages and disadvantages of cloning plants using cuttings, and a second table that shows the advantages and disadvantages of cloning plants using tissue culture. I'm just going to give you 3 minutes to complete this, starting now. Okay, that's half your time up. Try and get something in each column. Advantage and disadvantage of cutting, and at least one advantage and disadvantage of tissue culture. Great if you've got more than one for each column. This is a common 6-mark question, so it's really worth digging deep and seeing what you can come up with. Okay, time's up. So, did you manage to come up with something for each? For cuttings then, the advantage is it's quick, it's simple, it's cheap, and no expensive equipment is needed or no specialist knowledge is needed. Disadvantages, it's a lower yield, so fewer offspring are produced. Tissue culture. The advantage is you get lots of offspring produced because there's a higher yield. The disadvantage is this time more complicated, expensive, because it needs specialist lab knowledge and equipment. So I hope you managed to come up with something for each column. Well done if you did. Okay, these questions come up a lot in exam questions, and the reason they come up a lot is because they like to try and catch students out by mixing up the methods together and seeing if students have a really key understanding of the differences between each method. So sometimes they give you a diagram, as in the example shown. They'll ask you to look at that diagram to read all the information, and in this case, then use the words from the box to complete the sentences. So I'm not going to give you too much information at this stage, but I am going to give you 3 minutes. You should be choosing something from the box to complete the 4 sentences and then having a look at part B and seeing if you can come up with one other way of producing plants that are identical to their parents. So 5 marks for this question. Let's see what we can come up with. Time starts now. Just to avoid any confusion, the answer to part B isn't from the box. The box is only to give you the answers to the paragraph for part A. Okay, you've probably had enough time, so let's just read through it. At the top, we had pieces of stem being cut out from the plant. They were pushed into damp soil, and the new pieces grew into new plants. So very simple, no lab involvement, no tissue samples. So this is an example of just taking cuttings. And then it asked you to use the words from the box to complete the sentences. The colour and shape of the leaves are known as characteristics. The information for leaf colour is stored in parts of chromosomes called genes. The new plants are known as clones. And in this example, the new plants have been produced by asexual reproduction. Then part B went on to ask you to name one other way of producing plants that are identical to their parents. Well, the example you've been given is taking cuttings. So the other way you could talk about is tissue culture. So well done if you got 5 marks. Brilliant if you got 4. If you made any mistakes, you might want to pause the video, just have a read through and just check you understand where you went wrong. Okay, so moving on then to different methods of cloning. So we've looked at cuttings, we've looked at tissue culture in plants, but now we're going to look at animals. And this picture I briefly mentioned at the start of the lesson. This is Dolly the sheep. Now, Dolly the sheep was the first ever large mammal to be cloned from the cell of another adult. She was born in 1996. She was the only success after hundreds of attempts. And the method of cloning is called adult cell cloning. This cloning method is tricky and it's unreliable, but scientists do hope that it becomes easier in the future. Adult cell cloning is sometimes called fusion cell cloning. But for the purpose of GCSE, we're going to stick to adult cell cloning. What happens is they take tissues from a cell of a donor animal. And those cells from the donor animal are maintained in the lab and allowed to be kept sterile and secure, but they don't grow or divide. What they do is they then extract the nucleus from one of these body cells or tissue cells from the donor animal. They then involve a second animal, obviously of the same species, a female animal, and this time they extract some egg cells from this animal. They then remove the nucleus from the egg cell and insert the nucleus from the donor animal. So they would then have an egg cell from the second adult animal, which contains the nucleus from the first donor adult animal. Then that new cell, that egg cell, which contains the nucleus from the donor animal, is then given electric shocks and forced to fuse and multiply and develop into new cells which are identical, and eventually an embryo would be formed. That embryo is left to develop for up to 7 days, and that embryo is then implanted into a third animal, which is the surrogate mother. And that surrogate mother would then carry that embryo to full term and it would be born as a normal offspring. But what you would end up with is a cloned animal which has the exact DNA of the donor animal to begin with. So thinking about the diagram that I've just talked you through, I would like you to look at these 5 statements, which are the steps of adult cell cloning, all mixed up, and put them in the correct order. And you might want to number them 1, 2, 3, 4, 5 and just write the numbers down. However, it would be a useful paragraph for you to have to summarise adult cell cloning when they are in the correct order. But I will give you some time on the next slide when you get the answers to write it out in full. So I'm just going to give you 1 minute. The numbered statements are all mixed up and you need to try and get them in the correct order. Okay, time's up. If you need a little bit longer, just pause the video. Okay, the second method of cloning that we're going to learn about today is embryo transplants. Now, embryo transplants work differently than adult cell cloning. And in the procedure of embryo transplants, a developing embryo is removed from the pregnant animal at an early stage. So, fertilization has happened in the normal way. An embryo has been produced, but at the very early stages of pregnancy, that embryo is removed. And this is done before any of the embryo cells have a chance to become specialized. And then what happens is the cells that form that embryo are separated from one another. They're grown for a while in a lab in the best conditions, and they are transplanted back into host mothers. And all of those offspring will then be genetically identical to the original embryo that was removed from the pregnant animal. So if you look at the image on the PowerPoint slide, that embryo, 4 or 5 days old, could be split into about 10 different embryos, which would then be transplanted into 10 host mothers. So there's still going to be 2 parents involved because the embryo was fertilized originally in the normal way. But once that embryo has been fertilized and is starting to develop, it will then be separated. And this is the way that identical twins are formed. But in people, it isn't forced. Identical twins are formed when a very early stage embryo, due to some mutation or some mistake, does split in half. Identical triplets, the embryo splits into 3. Identical quads, which are obviously very rare, the embryo splits into 4. So embryo transplants is just a way of forcing a species to have identical offspring. The difference between embryo transplanting and twins is that those identical offspring will then be transferred separately into multiple host mothers, which will then carry those embryos to full term and give birth to the offspring in the normal way. So as we've just done with adult cell cloning, there's 5 statements below, and they are summarising embryo transplanting. At the moment, they're in the wrong order. I'm going to give you 1 minute to rearrange your statements into the correct order. The next slide will have the answers on, and you'll be able to pause the video and get them down as notes. So for the next minute, just write down the numbers and decide what order they should be in. 1 minute, time starts now. Okay, time's up. So, the order you should have come up with. The first stage, a developing embryo is removed from a pregnant animal. Then, the cells in the embryo are separated. The cells are grown in a lab to give them a chance to divide and multiply a few more times. Then they are transplanted into host mothers who carry the embryos to full-term pregnancy. And the last statement should be several offspring develop in different host mothers. They are all genetically identical to the original embryo. So if you want to pause the video now, there are your notes for embryo transplanting. A bit more straightforward than adult cell cloning, but still some important things to remember, such as the embryo is separated and then the separate embryos are put into host mothers. And the key thing to remember is the offspring will all be genetically identical to the embryo that you started off with. So this diagram summarises what happens in the case of embryo transplants. So you've got cow S at the top. Her eggs would be fertilised by sperm from the bull. Sexual reproduction, normal method in cows, and an original embryo would be produced. But the cells would then be taken from that original embryo, and in this example, that embryo is split into 4. It could be more than that. Those cells are allowed to develop into new embryos in the lab up to 7 days, and each new embryo would be transplanted into a surrogate or host mother. And the host mothers would give birth to calves. And all of those calves, all of those offspring would be genetically identical to the original embryo. But in this example, they are not genetically identical to either parent because there is still a mix of gametes, a mix of genetic information during sexual reproduction. So, just want you to look at this diagram, read through the labels, and just tell me, why are the calves born genetically identical to each other? And I'm just going to give you 1 minute. Okay, time's up. What did you come up with? So the reason they are all genetically identical to each other is because they were formed from the same original embryo. So still the sperm and the eggs from the same two parents, but that original embryo was just split. So why are the calves born to the host mothers not genetically identical to cow S? And I'm just going to give you 1 minute again. Okay, what did you come up with? So the reason they are not genetically identical to cow S is because the calves will have some genes or DNA from the bull, and they'll have some genes and DNA from the cow, the mum. Or there's this idea that sexual reproduction produces variation because the gametes mix together. The genes from each gamete are mixed together. Okay, so we're just going to finish off with a series of true or false questions. Each time I'm just going to give you 5 seconds just to make a note of T for true, F for false, and let's see what we know. So humans have 46 chromosomes in body cells. True or false? Okay, it's true. 23 pairs of chromosomes in each body cell makes a total of 46. The only exceptions to that rule are the gametes, because they have a single set of chromosomes, so they would just have 23. Next question. Gametes have 23 chromosomes. True or false? Okay, I made this one quite easy for you in the question 1 explanation, didn't I? This time it is true. They are the only cells in our bodies that have a single set of chromosomes. Okay, the male gamete is called the sperm cell. True or false? Okay, easy that one, isn't it? Yeah, it's absolutely true. The male gamete is called the sperm cell. Okay, so the female gamete is called the ovary. True or false? Okay, this time it's false. The female gamete is released from the ovary, but the female gamete is the egg, sometimes called the ovum, O-V-U-M. Going back to the male gamete, the sperm, that is produced by the testes. Sometimes you can be asked which organ releases the female gamete. The answer would be ovary. You could be asked the name of the female gamete. The answer can be egg. Which organ releases the male gamete? Your answer would be testicles or testes. And the name of the male gamete would be sperm. So some quite easy marks there. Okay, next question. Genes are small sections of DNA that determine a particular characteristic. True or false? Okay, if you got true, well done. Genes are a small section of DNA that determines a particular characteristic. So we'd have genes for hair colour, skin colour, eye colour. And a gene is a short section of a chromosome made up of that DNA. So don't get those words mixed up. Chromosomes are much bigger than genes, and they are all made of DNA. Next question. DNA is found in the cytoplasm. True or false? Okay, if you got false, you're absolutely right. DNA is found in the nucleus of cells, not in the cytoplasm. The function of cytoplasm you need to be aware of, it's where the chemical reactions take place. But DNA or genetic material or chromosomes are all found in the nucleus. So that question was false. Plants can only reproduce asexually. True or false? Okay, if you got false, well done. Some reproduce asexually, some reproduce sexually, and some actually carry out both forms of reproduction. Next question. Asexual reproduction means the offspring can look different. True or false? Okay, if you got false, well done. Asexual reproduction means the offspring will be genetically identical. There will be clones of each other. Next one. Taking cuttings is more expensive than tissue culture. True or false? Okay, if you got false, you were absolutely right. Taking cuttings is quite cheap. You can do it in your back garden. You can do it for houseplants. Tissue culture requires a lab involvement and tiny microscopic samples of plants to be taken. So tissue culture is more expensive than cuttings. It's the other way around. So that statement was false. Tissue culture is often done in a lab to make sure the conditions are sterile. True or false? Okay, that one is true. We want the conditions to be sterile because we want to be sure that only the plants we are cloning are going to be on that petri dish and are going to then develop into the plantlets. So that statement is true. So, to summarise the 4 modern cloning techniques. Number 1, cuttings produces many identical new plants from cuttings of an older plant. Tissue culture uses small groups of cells from part of a plant. Adult cell cloning transfers the nucleus from a body cell to an egg cell and transplants it into a surrogate mother. And embryo transplant is the splitting of embryos before they specialise and transplanting them into host mothers.